Start with the name of Allah Almighty, who is most beneficent and most merciful. My name is Sudhas Aruj, and I am from University of Management and Technology Department of Life Sciences. Today, I am going to show a presentation in this video about the nuclear injection. Before we going to work of micro injection, I introduce about the importance of micro injection. First of all, I show you a contents of my video. First, introduction, secondly, role of microinjection, and thirdly, gene transfer techniques, and fourthly, conclusion. And first, I explain the introduction of micro injection. 100 of years ago, Dr. Marshall A. Barber proposed a new technique, the micro injection technique. Micro injection process of using a glass micropipette to insert microscopic substance into a single living cell. First point is single living cell. It is used to create transgenic organism by injecting genetic material into the nucleus of a fertile oocytes. It is the most beneficent, and it is introduce of DNA into the oocytes, eggs, embryos of animals and other fertilization expression and analysis. Is the basic and commonly example of this method is used to study gain of function of a gene in mouse. Transgenic mouse technique, known as transgenic mouse techniques. And then I come to procedure of microinjection gene transfer. Basic steps are followed: insertion, transformation, expression, percentage of offspring contain foreign DNA, and expression. First of all, I tell you why microinjection technique is used. Because due to amphiteric nature of the phospholipid bilayer of the plasma membrane, polar molecules such as DNA and protein are unstable to freely pass through the membrane. So various physical or mechanical methods are used to overcome this and aid gene transfer. One of the method is microinjection, and some other methods are also. First is electroporation, second is microinjection, fourth is particle bombardment, third is sonofluorescence, last is laser induced, and fourth is beat transfection. And one of the microinjection partner is efficient use to transfer the gene into the organism. First is insertion. And the use of micromanipulator, normally performed under a specialized optical microscope setup called a micromanipulator. So use a micromanipulator, insert a gene into the another organism. So the target gene is placed in a container. A holding pipette is placed in the field of view of the microscope that suck and hold a target cell at the tip of pipette. And then second point, transformation. This pipette come to contact with membrane of the nucleus and injected through the membrane of the cell to deliver their content of needle into the cytoplasm, and then the empty needle is taken out. And the third step is then calculate the percentage of offspring contain foreign DNA, and approximately through this technique 10 to 30% of the offspring are contain foreign DNA. And the fourth one is expression. How DNA work, how gene work in the organism, and results perfect or not. And the next slide show advantages and also disadvantages of this technique. First, advantages of this technique is increasing the chance of integrated transformation, damaging. And the second is the small structure can be injected containing few cells and inject only a single cell through the per injection. And it is a direct physical approach, host is independent, and introduction of the target gene directly into the single cell, only a single cell, no one and other cell, no more cell required. And the third one is identification of transferred cell, injection of dye along with the DNA. Easily identification cell by injecting dye along with injected DNA. And fourth one is it can be used for creating transgenic organisms, particularly mammals. And the fifth one is no any antibiotic resistance and herbicide resistance marker is used for this technique. Advantages are given, but also some disadvantages are. First is injection can cause damaged embryonic survival and cause high mortalities. And this procedure is time consuming. Only a small numbers of cells can be treated. Means only one cell is targeted per injection. Means only a small number of cells can be treated. And the next types, 4 which is I show procedure, why injection are for membrane of the nucleus, nucleolus, and injection of needle through needle and the changing in the membrane in nucleolus, and successful result are given for insertion. And the last one is pictures show transfers of the data into another organism and show transgenic organisms. Conclusion is that this technique provides directly nuclear delivery of DNA, avoiding the endogenous pathway, and also ensure that the DNA is delivered intact. DNA delivered in this manner, and it must be very pure and need a lot of preparation as necessary to avoid fragmentation. And fragmentation of the DNA also protected by suspension in a high salt buffers. And this technique is very crucial and very important for converting the DNA into another organism. And also gene transfer method. Thank you for watching, and any question, please start.